In this video, I'm going to be walking you through the citric acid cycle. Now, this also has a few other names, for instance, the Kreb cycle, the tricarboxylic acid cycle, as well as the TCA cycle. And you'll hear me use these terms interchangeably throughout the video. So on our right side of the screen, we can see a couple of mnemonics and things that may be helpful to orient you as you're going through this process, where several of these were highlighted in a video for reference on Canvas, where the intermediate names can be represented by "our city is kept safe and sound from mobsters," where this indicates the 8 metabolites that we're seeing as part of the actual circle in this cycle. And to help you with keeping track of which reactions are irreversible, those are 1, 3, and 4. And the way that I remember that is by adding those up gets us back to 8. You may also find it useful to remember that glycolysis has irreversible steps, and 2 of those are also 1 and 3. In this right column, we see a few terms that are related back to the Cinco de Mayo mnemonic. Now, for me, that falls apart after a few steps, but you may be able to relate back to this as we start off the pathway. So getting right into it, we are starting off with oxaloacetate, which we saw during the process of beginning gluconeogenesis. And that has 4 carbons that are going to get added to acetyl-CoA, which has 2 carbons, by the enzyme citrate synthase. So that syn is where that first part of Cinco de Mayo is coming into play. And so we're going to see that water is going to be used during this reaction for the ultimate release of reduced coenzyme A. And as the name suggests, citrate synthase, we're synthesizing citrate. So our product here is going to be the citrate molecule, which has 6 carbons. And I kind of start off by drawing that big E, capital E. And if you've drawn with me before, you know I love to notice patterns in structures. So on the left stem of the E, we've got an alcohol. All of the other stems on the right side have COO minus. So those carboxylates are on the right. And this is our product citrate. So at this stage, we've got our as oxaloacetate and C for city as citrate. Now you'll notice that acetyl-CoA is not included in this mnemonic, and we've highlighted how that gets synthesized using the pyruvate dehydrogenase complex, which is the process of catabolism under aerobic conditions from pyruvate. So there's another video to walk you through that process if you need a refresher. So what are we going to do with citrate now? Citrate is going to get acted upon by the enzyme aconitase. So that's where that CO comes in for the Cinco de Mayo mnemonic. So this is a reversible reaction that is actually carried out in 2 steps. And so your textbook shows you a reaction intermediate that I'm not asking you to provide. Essentially, this reaction involves a dehydration that is followed by a rehydration. So if we look at the net here, we are using and releasing water. So you do not have to show this, but it may be useful if you're trying to remember what is happening in this reaction. And so another thing that I've highlighted in class is that the con term here may be useful for you to remember that we use water, we release water, they essentially act against one another. And so that is going to allow us to produce isocitrate isocitrate. And so the difference here is going to be the transfer essentially of this alcohol for this hydrogen, because we formed an alkene intermediate. So what does isocitrate look like? We've just switched that alcohol position from the center of the E to the bottom of the E. and that is isocitrate. So that is where our I comes into play. Isocitrate is going to have a dehydrogenase that is going to allow us to produce alpha ketoglutarate. So we are going to see an irreversible reaction, reaction 3, that is carried out by isocitrate dehydrogenase. And to save ourselves some time downstream, we have a lot of dehydrogenases in this pathway. I'm going to define DH as dehydrogenase so that you can also use that as you are learning your reaction mechanisms and reaction schemes. So in this process, dehydrogenase is hopefully jogging in your memory that we are removing hydrogens. And so the hydrogens that we are going to be removing are going to be donated to generate a reduced electron carrier. So that can be either in NAD plus or NADP plus. And the only difference between these two is whether or not a phosphate is present. So I'm just including that P in parentheses to show that it could be there, it could not be there. But that's going to produce either NADH and a proton or NADPH and a proton. So through this process of removing hydrogens, we're going to see this oxidative decarboxylation. The oxidation is where we're going to see the carbonyl formation. So we're going from an alcohol to a carbonyl, and the decarboxylation is going to ultimately come from the release of this carbon dioxide. And so what does our alpha ketoglutarate product look like? So we are releasing 1 carbon, so that is giving us a 5-carbon species. I'm going to show that there are hydrogens left behind after that decarboxylation. And we're going to have our oxidized alcohol to a carbonyl there, and our bottom COO minus, our carboxylate. And this is called alpha ketoglutarate. So that is where our K comes in. Alpha ketoglutarate is acted upon by another dehydrogenase to make succinyl-CoA, our first S product. So alpha ketoglutarate is going to get recognized by the alpha ketoglutarate dehydrogenase complex. And I'm again going to use DH as our abbreviation. We're going to see a few coenzymes as part of this process. One of those is coenzyme A in the reduced form. We're going to make our second reduced electron carrier here. So we're making NADH and a proton. And we're going to again have an oxidative decarboxylation, where in this reaction, we're losing this bottom COO minus as carbon dioxide. So what is our resulting product now? We're going to have succinyl-CoA, where the top of the structure stays the same. We'll see 2 hydrogens at this stage as well. The carbonyl on the left side hasn't changed, but now we have coenzyme A that is attached. And so this is succinyl-CoA, which is represented by safe in this mnemonic. Succinyl-CoA is going to get recognized by a synthetase, not to be confused with a synthase. And so there are many different coenzymes that are part of this process. So I'm going to leave generous space to illustrate that here. So the name synthetase is triggering that we are going to have a nucleotide triphosphate as part of this process. So we've learned that ATP and GTP can be interconverted reversibly. So I'm just going to show either one of those as well as either ATP or GTP as our product because of the activity of nucleoside diphosphate kinase. And so that is a great source of energy for other downstream reactions. And we're also going to see the release of coenzyme A in the reduced form here. So now if we think about the reverse reaction, we're just reversing the arrow for each of these steps. One thing I'll also note is that this is an example of substrate level phosphorylation, where we're going to see the addition of that phosphoryl group to succinyl-CoA to ultimately allow for the production of succinate, which is where we get our second S from. So this reaction is catalyzed again by succinyl-CoA synthetase, not to be confused with synthase. And as I briefly suggested, that is going to allow us to produce succinate, which looks like a pretty simple molecule here. Our 4 carbons arranged like this. And so essentially what we've done to change this structure is that we have this coenzyme A that is now released in this reduced form as a result of substrate level phosphorylation. Now we're going to see succinate that gets recognized by another dehydrogenase. And hopefully that is making you think hydrogens are moving around. So this is going to allow us to make F, which represents fumarate. And so this is going to be recognized by succinate dehydrogenase, where this is a step where those hydrogens from the dehydrogenation are going to be used to make another reduced electron carrier, where we're going to go from FAD to FADH2. And the reverse is true if we're looking at the reverse reaction. So now that we've had the activity of succinate dehydrogenase, we've taken succinate to fumarate. And so what does fumarate look like? We have a molecule that has a double bond between carbons 2 and 3. And so that is the result of our dehydrogenation, where we had a hydrogen here and a hydrogen here, essentially, that are removed to give to FAD so that we make FADH2. And this trans configuration is important for this structure, so you'll want to indicate it in the trans configuration. I'm going to move this molecule down just a little bit because we are getting toward the end of our Krebs cycle drawing. So now fumarate is going to get recognized by the enzyme fumarase. And this is another step that is reversible. That is going to involve hydration in this direction, forward direction, or dehydration in our reverse direction. And so this is going to allow us to produce malate, which we have seen also in the pathway where we're looking at pyruvate going back up to glucose. And so what has happened here? We have used water to allow us to hydrate the double bond. So that gives us our product, which is malate. And last but not least, how does malate M go back up to the start of the cycle for oxaloacetate? Malate is acted upon by malate dehydrogenase. And this reaction with the dehydrogenase should be triggering. Hydrogens are moving around. So we're going to see NAD plus going to NADH and a proton from this malate dehydrogenase reaction in the forward direction, and the reverse will also be true if we look at the reverse reaction. And so that now is just demonstrating the oxidation of this alcohol to our carbonyl that we had as a starting material. So this represents our overview of the citric acid cycle, where at this next reaction, this next term, all we need to do is combine this methyl group and that carbonyl carbon to make citrate all over again.